Hi everyone, this is Russ Altman from The Future of Everything. This week we are resharing a conversation I had in 2021 with Joseph DeSimone, a professor of chemical engineering at Stanford University. This one is about health. Joe tells us how 3D printing is transforming healthcare. His group is using it to make vaccine delivery easier and more effective. They're also creating implantable chemotherapy that kills tumors while having less side effects for the patients. I hope you'll enjoy this glimpse of how 3D printing technologies are being used in novel and unexpected ways. Before we jump into this episode, a reminder that if you enjoy the podcast, please rate, review, and follow it. That will help us grow our listenership, and it'll make sure that you're never surprised by the future of anything. The last few decades have witnessed a revolution in the field of 3D printing. Now, this is the technology that allows us to create 3D objects based on a digital model that starts out in a computer. These usually work by having soft materials of some kind that can be deposited or formed, often layer by layer. And as that material hardens, it can create a variety of shapes, including complex geometries that include holes and support structures, and really quite complex 3D structures. These 3D printing technologies have advanced with the introduction of more diverse materials, more precision in the creation of these objects, and we have seen them used to create a wide variety of things, including rapid prototypes of new products from spoons to complex devices with multiple parts. They can often be used to replace difficult to find parts for machines. They've even been reported to create working guns. But that's not what we're talking about today. We're talking about something we don't usually think about, which is how can these 3D printed objects become critical in healthcare? In fact, 3D printing has promise in providing new ways to deliver therapies, including medications and vaccines. Professor Joseph DeSimone is a professor of radiology, chemical engineering, chemistry, and operations information and technology at Stanford University. He works on the development and use of 3D printing for applications in health. Joe, how has the evolution and improvement in 3D printing created opportunities for health?
First of all, Russ, it's great to be here with you. Thank you. So what's happened, 3D printing has been historically a prototyping technology. And it's allowed an amazing ability for designers in a lot of different fields to design new products. The big transition that's underway is that 3D printing, the technology itself, the printers, the hardware, the materials, the software has gotten to the point where 3D printing can now transition to manufacturing and scalability. And when you start thinking about one's anatomy, the bespoke nature of perfectly fitting products and scaling at the same time is a really interesting intersection for the field.
Great. So I know that you've been very involved in something called clip technology for 3D printing, and you've been involved in companies, and I think this is a basis for a lot of what you do. Can you just give us a high-level description of how this technology works, and maybe some of the sense of its capabilities and where the frontiers are?
Yeah, so 3D printing was invented in the 1980s, and it emerged out of the discipline of mechanical engineering. Where one very precisely deposited layer by layer films, like a Xerox machine. But you do it over and over again, pretty soon it gets thicker and thicker and thicker. And that's what 3D printing was.
Right.
And it's really been 2D printing over and over and over again.
Because you're kind of layering these printouts, so to speak, on top of one another to create the shapes.
Right. That's right. And people are probably more familiar with like a MakerBot kind of product, which is like a hot melt glue gun rastering across the surface.
Right.
And my team and I came at this from a chemistry point of view and a physics point of view. And, chemists like to grow crystals. And we were thinking about, wouldn't it be great if we could grow these products continuously? And we invented a method that uses light and oxygen to grow products that allows us to go to 25 to 100 to even 1,000 times faster than the traditional approaches.
Interesting. And so this is when you say crystal, you mean that you're getting the material to not be so much layered, but actually form a three-dimensional. Is there still layering? I guess is my question.
So there is it's more like playing a movie than taking snapshots.
Okay.
Yeah. So there's individual frames, but now they're happening rapidly, so rapidly it looks like a movie.
Okay.
And the objects are really growing. So it actually looks like if you've seen the Terminator 2, a T-1000 coming out of a puddle.
Right.
Right. We in fact envisioned, could things emerge out of a puddle of resin? And if we could do that, and could the mass of the object be derived from that liquid coming from underneath it? Could we grow that very fast? And can that liquid emerge into a material that had the properties to be a final part instead of a brittle plastic, which was typical of 3D prototyping?
Great. So before we dive into health, one more question. How precise can we engineer these? Like what's the plus minus on the sizes? Because you made a reference to, we might be able to make bespoke, just made for you devices for individual patients. So how precise are we? And then what is the range of materials that can be used with these technologies?
So, precision, it depends on what you're making, what's needed, right? And so from a running shoe to a football helmet, the printers there have a pixel size of 162 microns. So
that's a millionth of a meter.
It's 0.16 of a millimeter, right? 162 microns. It's almost 20% of a millimeter. And then when you get into oral health and things in the mouth where you can fit is more demanding, we drop to 75 microns.
Okay.
Right, which in a human hair is 100 microns.
Right, so we're talking human hair.
Right, we're talking a fraction of a human hair, but we're now down to building a printer at single-digit microns, and a red blood cell is 8 microns in diameter. So we're going from the macroscopic down to the cellular length scales.
Great. And what about the material, the range of materials? Are we looking at metals, or are we still thinking plastics? Where is all that?
It's all polymers.
Yeah.
We've had a breakthrough in how to do this with polymers. So polymers have properties that range from high performance engineering materials at 4 gigapascal with stability out to well over 200 degrees Celsius. To things that look like engineering plastics that would be part of the dashboard of a car, or the support products for a battery in an electric vehicle, to dentures and replacement of things in the mouth, all the way to elastomers. That would be great for if it's energy returning, like a running shoe, you want to bounce. We have elastomers that do that. We also have elastomers that dampen energy and absorb impact. And then we also have water-soluble, degradable, and biodegradable kinds of products for sustainability reasons, but also for biodegradation in the body. So the whole gamut of polymers. We don't do metals. Somebody needs to have an invention in metal 3D printing like we've had in polymers.
Yeah.
I don't know what it is, and I don't see it on the horizon yet, but we're just polymers.
No, that's very impressive, because you mentioned teeth, which obviously have to be very hard, and then you mentioned running shoes, which obviously, so this is the future of everything. I'm Russ Altman. I'm speaking with Professor Joseph DeSimone about the future and present of 3D printing. Okay, let's go to health. I know that you're working on vaccines. Vaccines have been on everybody's mind. What's the problem that needs to be solved, and how is your team approaching it?
Yeah. So, well, first of all, we're at an amazing period in history where mRNA-based vaccines have taken off, and it's really just the tip of the iceberg. mRNA is you're going to see that happening in lots of different areas, and it had a long road from silencing RNA to, all sorts of delivery approaches in oncology, but it's our moment. It's the moment for the field in vaccines, and it works, right? And all the nano carriers that were developed for oncology, which really didn't pan out a whole lot, work really well in infectious diseases. Now, the interesting thing is that needle, well, first of all, the vaccine's in a liquid formulation, and we've all been hearing about refrigeration and low temperature storage.
Never has society cared more about refrigeration than it has in the last year.
And if you're starting a new lab and you're trying to buy a refrigerator right now, it's really hard because every other place is a back order right now. So that's a separate topic. But low temperature storage is really important for these carriers. That's a problem, not only here in the United States, but globally, it's a bigger problem. And then you think about dealing with malaria and measles and global access, it's even a bigger problem. That's one thing.
Yep.
The other thing is the cargo, the vaccine itself went deep into our arms. It's a minor needle, but it went into the muscle. And we have far more immune cells and the targeted cells just under our skin, in the skin, than we do in the muscle.
Huh.
And so we've been developing microneedles that are 3D printed. Now, microneedles have been around for about 15 years, and it's a way of delivering molecules just under the skin, into the dermis. Most of these microneedles have been fabricated using a technology from the computer industry, used to make chips. And they were making microneedles, and they showed that this is a valuable area of delivery. But the challenge is the kind of structures you can make in a fab are limited in geometry, but they're also really limited in materials.
Right.
So interestingly, we figured out that our breakthrough in 3D printing, which is continuous, uses light and oxygen, works really well at this microscopic level. And now we're printing 3D printing microneedles. And our microneedles can have geometries that you can't access in a fab. And we can make them out of materials that you can't access in a fab.
And when you say fab, those are the fabrication facilities for like silicon wafers and other IT type products.
Yeah. Yeah. And so we can make them out of what are called soft materials instead of hard materials.
Yeah, you just described an amazing repertoire of capabilities in terms of the physical properties.
And what we now do is we can now use those to deliver these vaccines. And so we've got a paper under review right now at the Proceedings of the National Academy of Sciences for a delivery of protein-based vaccines, the old type of vaccines, to the dermis, and we see 50 times more antibody response, delivering the same vaccine in a more precise way to the dermis than you do from a needle, 50 times. Now,
so would the idea be eventually to get more out of a single vial of a vaccine, or to get a better response, or both?
It's both, and it's more than that too. So not only you could do dose sparing, you can give somebody 1/5, or you can vaccinate 50 times more people, right, with the same amount of material. But
I just want to bookmark that, because that is a huge, right now in the world, that would be a huge capability.
And then here's another one. The immune response, there's two big channels of immune response. There's Th1, Th2, meaning the antibodies versus T cells, the long-lived T cells. We see a much more balanced response by delivering this into that dermis layer than you do into intramuscular delivery. So which should be a more long-lived, robust response, because it's both T cell and B cell response. And more interestingly, as important, the microneedles, the active ingredient now is in sort of like a lyophilized form of the active ingredient. Instead of in liquid form, it's a coating of sugar, actually, on the microneedles themselves as a patch, kind of like a Band-Aid.
Yep.
And it's a solid state, and things in a solid state are more shelf stable for long periods of time.
So that can take a plane ride and get dropped by a parachute. Yeah.
So here's what this could mean. We think that microneedles could be the OS of vaccine design, could be the operating system that could, in a plug and play approach, could address any antigen in time of need. It could eliminate the cold chain challenges. It can eliminate the healthcare workers' need for administration. Right now, there's 300,000 healthcare workers trying to administer all these vaccines, right? And we think it's scalable to billions of doses. So we think it's a new approach that could really address a lot of the supply chain challenges, the logistical challenges, and it's a better way of a better immunological response.
So the vision, just to make sure I have this right, the vision is that these things come in, they're like patches, but they have little tiny needles, probably almost barely perceptible, but so, and then you might be able to slap them on your arm or slap them someplace, and that will start the delivery. There may be less or no healthcare workers involved. And is that the vision?
And it could be, you could get it in the mailbox.
Yeah.
Right. And UPS would, or Amazon would deliver it, or Walmart would deliver it to you, or you'd go pick it up. And yes. And so this would allow us, for example, the Gates Foundation is trying, wants to eradicate measles now, just like polio. Mm-hmm. And you're going to need to eliminate the cold chain issues and the sharps. And right now there's shortages of vials and all these things. And so it's really getting into a solid state delivery approach for these important vaccines.
So I know another one of your great interests and expertise is this process of translating from discovery in the lab to actually something that can scale and really impact millions or even billions of people. So where are these ideas in that spectrum of early idea to we're ready to ship? And what are the big logistical things that you have to overcome in order to be able to deliver this? I'm sure people are wondering, when can I get this? It sounds great.
Yeah. So we've been down this road in a not as important area, but an important area. So think about dentures, right? That's a $14 billion market. When you get fitted for dentures, it's typically 8 chairside visits of routine, going in over and over again to get it perfectly fitted because it's handcrafted.
Right.
We are now working with others that, have digital scanners for the mouth. Right, just like your iPhone, but and it scans through videography, right, and close cousin to that. And it's a digital capture. So we've now developed the world's first FDA-approved 3D printed dentures, and it took us about 2 years to do that. But you think about what's happening. You now go to two chairside visits instead of 8. That's
huge.
It's perfectly fitting instead of a person, thinking about, putting that blue tape in there. Is it rubbing here? Is it rubbing? That's archaic.
Now you're giving me post-traumatic stress just thinking about my last trip to the dentist. Yes.
So this is digitally fabricated now with precision that you can't replicate with handcrafted things. And if you're trying to scale anything that's bespoke, you've got to do it digitally with digital scale, So we've been able to accomplish this. And these kinds of materials, the denture base, the denture teeth, are close cousins to the kind of sheet of plastic that we'd have on the patch for delivering vaccines.
Right.
So we're now going through collecting all the data in animal models for the vaccine approach. And we're working with industry, all the big names in the vaccine space, for using their vaccines and delivering them. They're coming to us now because they want to get away from the cold chain issues. They want to go to dry patches. They think this is going the way of Amazon.
Right.
But what I think this is also lending us to is wellness monitoring, which, look, Tom So and our colleagues here at Stanford have been looking at ways of monitoring the human body, like people monitor jet engines, and all the sensors. And, the readouts of glucose, is a really important one, or oxygen is really important. We only monitor 2 molecules like that. And so the idea of, can we use these same microneedles to access interstitial fluid? Because that's where we're delivering.
So you turn it and you pump it in the other direction out of the skin to get a sample.
Yeah, now people have been obviously looking at blood all the time for this. But, we have 5 times more interstitial fluid than we have blood. And the molecules found in interstitial fluid mirror the molecules that we see in blood.
Right.
Way better than tears or sweat does, right? And so the ability of having new kinds of devices for monitoring, again, 3D printing opens up these kinds of objects, especially to get down to single-digit micron resolution, for new types of sensors in that area. So this is where this field's going.
I know that, in fact, you mentioned mRNA, and mRNA has also been very successful with the vaccines, but it has not been so successful in its applications for drug delivery, for example, chemotherapy for cancers. I know that you've also been working on these 3D printing technologies for the delivery of drugs. Where is that? What's the idea there, and what's the status?
So one of the big challenges in oncology in particular is the systemic side effects. And what's also interesting is, imaging and early detection has taken off as a field, but a lot of our treatments are still systemic when the cancers are locally identified.
Right, you only have cancer in the lung, but I give you this poison that goes throughout your entire body.
It's crazy. It's crazy. So the opportunity for local regional delivery is increasingly important. And X-rays do that. X-ray treatment is very local, it's focal, right? But chemotherapeutics and other molecules are not. And so the opportunity for doing that is high. And so, for example, I mentioned that the mRNA delivery in oncology was challenged by the nanocarriers not getting it into the different barriers that, and biological barriers for, the stroma blocking chemo, small molecules, let alone big nanocarriers, can't get into these tissues. So we're taking a much more engineering response. People have been using polymer-based drug delivery devices. Bob Langer pioneered the gliadal wafer in brain cancer. But now we're starting to design advanced microfluidic-like devices for local delivery. And sometimes they're assisted by electric field-assisted delivery, something called iontophoresis, where you can have an electric field drive molecules into poorly vascularized tumors. So we're building devices that can drive drugs that have not been accessible through IV injection for local delivery. And we're doing it in pancreatic cancer. We've had a really great data that shows that we can deliver molecules like gemcitabine into these tumors. And normally when people get pancreatic cancer, those tumors are very slow growing. They typically started 20 years before they were symptomatic. And it's often too late. They're locally advanced. And they often wrap around big blood vessels that make surgical resection very challenging. Well, we've been able to address some of that with this local delivery, and we've been able to show 40% regression of tumor volume for these very challenging pancreatic cancer tumors. The first time people have seen tumor regression. So we published a paper in Science Translational Medicine using these local drug delivery approaches. We just spun out a company called Advanced Chemotherapy Technologies, or ACT, that is funded by Vinod Khosla here in the area. But being able to drive these drugs into these tumors and using a new advanced 3D printing devices for doing that. So a very convergent approach with lots of different disciplines coming together.
So I just want to make sure I understand. So in the case of the pancreatic cancer, is there a surgeon who's implanting this device somewhere? So I just want to blow up this idea of local and how do you get it to be local? And then do you have evidence that in fact, these very potent cancer medications are only acting locally and you have much lower levels in the periphery that doesn't have the cancer?
Yeah, so in this particular case, yes, it's surgically implanted directly above the tumor in the device. There is a tube that comes out to the belly, PICC line.
Okay.
And that can be hooked up to a wearable for local delivery of chemo or at an infusion center. And the drug is not only, it goes to that device and there's an electric field that drives the drug deep into the tumor. And we can do it at a wide range of dosing, especially with the wearable, metronomic dosing. It's long, slow amounts of dosing.
Right.
And we are actually seeing 40% regression of the tumor. The drug concentration in the tumor is very high. The amount of drug in systemic in circulation is almost not detectable. So it's just the opposite. When you get an IV of these really potent chemotherapeutics, the amount of drug in your blood circulation is really high, and there's actually very little in the tumor.
Right.
Because it's blocked, and we have just the opposite.
So forgive my curiosity, but what does this look like? What can you just, using the miracle of radio, can you explain what this thing that's implanted looks like?
All right, it's a little bit bigger than a quarter, maybe about $50, if you remember those.
Yes.
And it's a little device that has a semi-permeable membrane and a little reservoir that's about 2 milliliters in volume, with a tube and a wire that comes out to the belly.
Yep.
And there's actually an electrode in that little reservoir. And then you basically tape another electrode outside the body opposite the device. And that's enough of an electric field gradient to drive the molecules into the tumor.
Great. And so that's a great example of, and now you can imagine wherever the cancer is, liver, pancreas, lung, that you might be able to come up with similar devices to deliver this highly concentrated chemotherapy, much more local than previous.
And so we've got a big focus on precision delivery using these advanced devices. You can imagine you can take someone's CT image or MRI of their tumor and actually make it bespoke there too. Right. For local delivery. So those are the fields we're going after.
So this is very exciting, and I did want to turn in the last couple of minutes. It's a little bit of a turn, but I think it's very important. You've also been involved in a lot of issues of diversity and trust in bioengineering and in medicine and healthcare. And while that's a great topic, you might not think that a person who's a 3D printing whiz would be doing this. So can you make that connection for me and tell me the kinds of things that how this impacts your work?
Well, everything we just talked about I think it was self-evident about the disciplinary diversity, right? In chemistry, material science, software, hardware, all coming together. And everyone gets that. But human diversity also brings a level of innovation that's really important. Diversity at its core is a fundamental tenet of innovation, whether it's disciplinary diversity or human diversity. And I've had researchers and trainees from all over the world. And when somebody grew up with not much money, they think about problem solving very differently than somebody who grew up with a lot of money. And that was evident to me early in my career. And that's socioeconomic diversity, gender diversity, experiential diversity is part of the canvas of our innovation for our group. And so I think I think the hallmark feature of our group is we're trying to do new things, trying to do innovation. And that requires diversity in a broadest sense to drive that. And so it's getting that right. And then what's also clear is you've got to be clear about your values. When you're clear about those values and you talk about that, then you can become a destination for excellence wherever it is. Mm-hmm. People know they can go there and feel included and can belong. And so, so that's a big part of it. Our group a long time ago, were part of a meeting that was going to be in South Carolina back in 2000 when the Confederate flag was hanging over the state capitol. And there was a boycott from the NAACP. And we honored that boycott and didn't go to the meeting. And then the chemistry community started talking about us, leading like that. And that story got out. And we became a destination for excellence because of those things. And so I think it's important to be clear about one's values and the role that diversity, both human diversity and disciplinary diversity, drive the innovation engine.
It's fantastic to hear. And that theme has come across in this series of conversations that I've been having with innovators. It comes up time and time again that they see the impact of their diverse teams, and it's not theoretical. It leads to faster progress on all the problems that they're trying to solve.
It's mathematical. You can just see all the different inputs coming together. It's like combinatorial approaches. And trying to cast the widest net in diversity, both disciplinary and human, is really important.
You have been listening to the Future of Everything podcast with Russ Altman. I want to remind you that the Future of Everything started out as a radio show on SiriusXM, so you'll hear references to that. Now it is 100% podcast, but we still have access to the great shows that we taped with SiriusXM. There are more than 215 of them, and they cover an extraordinary range of topics. Please remember to subscribe or follow the podcast on your favorite listening app, and also to rate, review, and tell your friends. You can connect with me on Twitter @RBAltman and with Stanford Engineering @StanfordENG.